All right, we're going to do part 2 of chapter 1, the microbial world in you. And this section of your notes really deals with kind of the historical impacts of microbiology. Who were some of the different scientists who contributed to the discovery of microbiology? And then we're also going to look at some of the different branches of microbiology and some of the applications. And finally, a little bit on emerging infectious diseases. You guys are kind of living through a time of an emerging infectious disease in COVID, but we're going to look at some of the factors that contribute to that as well. So first of all, when we look at how microorganisms are classified, Carl Weiss, I'm not sure if I'm pronouncing his name right, but he basically broke it down into 3 domains. We have the bacteria and the archaea. And remember, both of these here are prokaryotes, okay? And then the rest are the eukarya, or the eukaryotes. So that includes your protists, your fungi, your plants, and your animals. And remember, protozoans, the animal-like protists, and then the algae, the plant-like protists, belong into the protist category, and then your fungi. And then your animals, those were like your helminths, your microscopic worms, parasitic worms that fall into that category. So with a brief history of microbiology, some of the first people, we have Robert Hooke. You probably studied him briefly in biology. He was the very first to observe cells and actually use the term cells. He was actually looking at cork. So he was really only looking at the dead cells, with just the only thing remaining of the plant cell was the cell wall. So he described them as looking like little compartments or cells, and that's how we got that term cell. And then Anton von Leeuwenhoek was the first to really work on microscopes. He was a microscope maker, and so he was able to observe some of the first organisms, the first tinier organisms, because of the use of his microscopes. And so that kind of opened a whole new era of discovery in terms of microbiology once we had more powerful microscopes. But up until this point, there's a big debate over what was called spontaneous generation. The whole idea that living things can come from non-living things. All right? We use the term abiogenesis. Anytime you add a on the front, it means without. So in this case, without life, like living things came from non-living things. Biogenesis is the opposite of abiogenesis. That means living things can only come from pre-existing living things. And Rudolf Virchow, as part of the cell theory, is the one that said that the living cells can only arise from pre-existing cells. And so that's a big part of our cell theory. And so there's this big debate because, prior to this, people thought, that, mice just arose from piles of grain. Well, obviously a pile of grain cannot make a mouse. But, they thought that, the drinking water, suddenly, sprouted worms or things like that, where the troughs the horses drank out of. Well, there was probably some microscopic worms on the horses themselves or something that added to the water. But all these different things, you were, if you got sick, it was because, you sinned or something bad, you did something bad. They did not understand microbes because they couldn't see them. And so there are a lot of people who believed in this vital force that somehow forms life, that it just kind of spontaneously arises. That's why it was called spontaneous generation. So a bunch of scientists started doing a series of different experiments. Francisco Redi in 1668, he did an experiment with jars and meat. He was trying to show that flies do not suddenly just develop from rotting meat, that they don't arise from the meat itself. So he had 2 jars. He put some decaying meat in there in both jars, but he covered one up with some cheesecloth. So air could still get in, but flies could not get in. And what he observed is flies went into the jars that were open, and they laid their eggs, and then maggots developed. That's the larval stage of a fly, maggots. And so it developed from that, and in the jar that was covered, where the flies couldn't get in and lay their eggs, then no maggots appeared on that meat. And so he was basically trying to disprove that idea of spontaneous generation. John Needham boiled some broth, and then he cooled it, but the broth still got cloudy and developed microbes in it. So he just said, it must be spontaneously generating. It must be just coming from the broth and just spontaneously arising. What he didn't know is if he left the broth uncovered, there's obviously, mold spores and things in the air that dropped in. Lazzaro Spallanzani, he did broths heated after sealing and showed that they didn't have any microbes in them if you covered them up. If you covered up the broths and kind of put a cork kind of loosely in them and then boiled them gently, it killed off all the microbes and nothing grew in there. But then people accused him of, you didn't give him air. And air is the vital force that everything needs in order to have life in it. So Louis Pasteur, in his famous experiment where he used these S-shaped flasks, he basically did the same thing as Spallanzani did by heating up the broths, but he was able to leave one tube open so microbes could drop in. And in the other one, here's his little experiment here, he basically opened one here, and then he hooked up a S-shaped tube here. So there was still air that could get in, but the microbes just collected down here. They didn't get into this broth, so this broth stayed clear because he heated it. But then he did the same thing, and he busted off the top here, and then microbes dropped down in there, and then that one did get cloudy with microbes. And so his S-shaped experiment basically disproved spontaneous generation, that the growth in the broth did not just spontaneously generate, but it came from the air, that there was microbes everywhere in the air. Hopefully you guys remember what that means, that microbes are everywhere almost seemingly at once. Ubiquitous. All right, I had to throw that in there. So, biogenesis, life comes from life. Pasteur kind of put the nail in the coffin for abiogenesis. He said, no, living things have to come from living things. He also contributed to aseptic technique, the techniques that prevent contamination by unwanted microbes. Joseph Lister, Ignaz Semmelweis, all of them played a role in using aseptic technique. And that's any technique that helps prevent contamination. So when we wash our hands, when we wear gloves, when we flame our inoculating loops so that we don't add any microbes. When I put the broth and the augers in the autoclave to sterilize, that all kind of prevents that. Pasteur also did work with fermentation. Obviously, even back in his day, microbes were used to help convert sugars into alcohol to make beer and wine. They still do that today. But sometimes spoilage was occurring, and what Pasteur realized is what was causing the spoilage was unwanted microbes that were getting in there, causing that, and that just totally destroyed the beer and the wine. And so a lot of people were asking him, how can you help us with this? So even back then, he was both trying to use microbes to help make the product, but also to prevent spoilage of the product. Pasteur came up with the process of pasteurization, named after him, where you use high heat for a short duration. It kills most of the microbes, but not all. So anytime you drink milk that's been pasteurized, there's still microbes in your milk. There's still bacteria in there, but it's at such low quantities it's not going to do anything or harm you or anything like that, but it's still there. That's why milk spoils after so many days, even if you keep it in the fridge, because those microbes are still there. They're reproducing it, but at a very slow rate, okay? And then vaccination, Edward Jenner came up with the first smallpox vaccine. And so we're thankful for him because those vaccines have been improved over the years, and now we're all anxiously waiting ourselves to get the COVID vaccine to hopefully help slow down the spread of COVID, and so we can take control, a little bit more control. Another big idea that was covered in this time period was what we call the germ theory of disease. And this is the idea that particular microbes cause certain diseases. Okay, the germ theory. Again, remember, a germ is something we sometimes call a bacteria or some other microbe. And so the germ theory of disease basically states that a certain microbe will cause a certain disease. Okay, certain microbe will cause a certain disease. And so some of the people who did research on this, Ignaz Semmelweis, he looked at hand washing and how that prevented the transmission of what was called puerperal fever, or childbirth fever in moms that were giving birth to babies. And he was showing how if the doctors who were delivering these babies washed their hands in between and so on, it helped prevent the spread of that from one patient to another. So he was trying to show that, yeah, it's coming from the actual individuals that are doing this, doing these deliveries. Joseph Lister came up with a chemical disinfectant to prevent surgical wound infections, and again, sterilizing and kind of washing the instruments and washing their hands in this carbolic acid to kind of prevent the spread of disease during surgeries. And then the one we have to thank probably the most for kind of showing us the germ theory of disease is he basically came up with a series of steps to show that a particular bacterium caused anthrax. And so he did a series of steps called Koch's postulates, and it proves that a specific microbe causes a specific disease, and that is basically the germ theory of disease. How did he do it? He basically took a cow that had anthrax, and he took a tissue sample from it, and he isolated that microbe. He grew it out on a some sort of medium, and he isolated that microbe, that bacteria, and then once he had that isolated, he took that and injected it into a healthy cow. And if that cow came down and got sick with the same symptoms, he then took a tissue sample from that newly diseased cow, and he grew it in pure culture and tried to grow up those colonies, and then he compared the two. And if they were exactly the same, he could basically state this microbe causes this disease. And so we're very thankful to Robert Koch for kind of basically demonstrating, and we still use his steps today. That's still what doctors are going to use to kind of find the cause of a particular disease. All right. Some other individuals who kind of played a role later on in the golden age of microbiology, Paul Ehrlich in the early 1900s speculated that a magic bullet could destroy a pathogen without harming the host. And so he developed the synthetic arsenic drug salvarsan to treat syphilis. And then in the 1930s, sulfa drugs were synthesized. So we're kind of getting into those first almost drugs that can help treat disease. Antibiotics, of course, these are chemicals produced naturally by bacteria and fungi to actually kill other microorganisms. And so Alexander Fleming discovered penicillin. That was basically the first antibiotic. It was an accident. He was growing some bacteria in some petri dishes like we do, left a few uncovered, and of course there's always mold spores floating around in our air, unfortunately, and some of them dropped on his petri dishes. And wherever those mold spores grew up, there was this clear zone around the mold spore where no bacteria grew. So basically that mold was killing the bacteria, and that was the penicillium mold. And so that is what helped produce penicillin, which was mass produced in the 1940s, which they thought would be kind of the end-all be-all, but now we know that many microbes are becoming resistant to that, and so that isn't the end-all be-all. We still have to find some other things. I'm just going to stop right here because I don't think I have enough time to get all this done in one taping, so I think I'll stop, and then we'll pick up on the basic different branches of microbiology in the next video.